Hi, my name is Shania Bratcher. I'm an incoming second year or sophomore here at UNC Chapel Hill. I've been in the Armistead lab this summer, and it's actually been a blast. I've had a really great summer, as this is my very first time doing research, period. But today I'm going to talk to you a little bit about optimizing CRISPR-Cas9 gene editing to establish isogenic TP53 knockout in human leukemia cells. I know that was probably a mouthful, so I'm going to break it down to what's a little bit more familiar to everyone. So first off, before I tell you what is TP53, where is it? I'm sure many of you have seen this before. This is a karyotype. And humans typically have 46 chromosomes. When you divide that by 2, you get 23. You get one pair from mom, and you get the other pair from dad. Probably like, why are you telling me this? Well, TP53 is on chromosome 17, and I'm going to talk a little bit more about that on this slide. First off, what is TP53? Going back to the localization, again, this is on chromosome 17, specifically 17p13.1. But TP53 is a tumor suppressor gene, which is basically when you have cells that are overproliferating, meaning they're dividing way too rapidly, you have the guardian of the genome to say, whoa, let's put a halt to this. And it typically undergoes something called apoptosis, where it's triggering cell death. Humans have 2 copies of TP53, or typically should have. And that's, again, one from mom, one from dad. But specifically, most frequently mutated genes are in human cancers from this gene here, TP53, which is why we are using this to study for cancer therapies. All right, so what have I been doing this summer? We're talking about the scientific gap in TP53 mutant AML. The issue is there are TP53 mutations that drive clonal selection in AML, and AML stands for acute myeloid leukemia. So we took cells that were in a nitrogen tank, and they came from patients who had this specific cancer. And what we're trying to figure out is why are healthy cells not winning against this race between the mutant cells? And when we look at this here, this is called a Kaplan-Meier survival curve. And here's different genes or different types of genetic mutations that occur. And as you can see down below, we have TP53 aneuploidy as one of the lowest survival rates. And that is something that scientists are very curious about. So we're going to talk a little bit about that today. We're trying to figure out, are these mutations driven by dominant negative effects, loss of function, or gain of function? Loss of function, as it may sound, is literally TP53 losing its ability to stop. Think of this as TP53 being the brakes. When you're driving a car, imagine driving without brakes. Terrible, right? But what if I told you there's something worse than that? Gain of function is when TP53 or any other gene would gain these abnormal abilities. And what would happen is that's like you pressing on the gas about to approach a building. It's way more dangerous. Then we have dominant negative effects. Remember I told you we have 2 alleles, right? One from mom, one from dad. That's like you having a functioning one, and then your second allele is not working properly. And I know that I'm breaking this down a little bit, but I want my science to be a little bit communicable so people can understand. So what is our solution? This summer, we have set up CRISPR-Cas9 to create isogenic TP53 knockout clean slate models. And what that does is that we're going to use that to be able to isolate these mechanisms to clarify how these drive to therapeutic resistance. And specifically, I'm curious about this because there's something called relapse, right? So let's say a patient who specifically has leukemia, we'll talk about it in this context, and they're getting their treatment, their chemotherapy, but then they're getting better, right? They're going through remission, everything's going well, but then there's a tiny amount of cancer still left, and that's something called minimal residual disease. And what that is you have a small amount that's left over that ends up being resistant to these chemotherapies, and then that's when that ends up having patients go through relapse. And unfortunately, this happened with my mentor's patient recently, and so now they're in hospice, which is very unfortunate. So as a scientist, I really want to bridge this with the clinical context. So what I've been doing this summer, I'm going to talk about my methodology. It's my little picture. But like I told you, we took these cells out of the nitrogen tank and we grew them in media. We used RPMI and IDM, but we decided to use RPMI because we noticed our cells were dying in the other culture media. And first we used about 200,000 cells, recognized that wasn't enough, so we allowed them to grow to up about 2 million. And then we did something called electroporation, my favorite thing. Actually, no, not really. So electroporation is basically shooting electricity within your cells, and it allows the cell membrane to open a little bit just so you can get, in this context, our complex inside. So there's something called a guide RNA, and we also have something called the Cas9, which is actually making the cut. So we want it to lead it to the TP53, where that is on the chromosome, and we want the Cas9 to make the cuts. However, Cas9 is a really big protein, so that was a very hard thing to do. So we had to do some optimization. You guys know science. You got to change things up and do a whole bunch of different things to make sure that you can find something that's a little bit more quantitative and qualitative. So for our validation, we had something called a genotypic validation, which is PCR, which I'll show you my results in a few minutes. And then we also had our phenotypic one. We use GFP as kind of like a control, so we can see the cells if they were able to get the guide RNA and the Cas9 inside. And then I'll talk about this a little bit more. We also did Nuclein-3 treatment, but again, I said I'll talk about that a little bit more next. Results. All right. So this is my very first PCR, and it was a little disappointing, but hey, that's science, right? I was expecting to see 2 bands for TP53, right? In this case, I did not. And it basically told me that I did not knock out TP53. However, I did it again. These results were a little bit more questionable. I ended up getting bands, but they were for wild type. When we set it up for wild type knockout, there was not supposed to be a band here. So as a scientist, we had to regroup and said, what could have happened? So for one, mistakes happen. We could have mixed up the cells. We could have said, oh, wild type, knockout, knockout, and we could have had the knockouts first. But we did use AAVS1 as our control, so we knew that we weren't getting background noise, but we knew for a fact that we would have to redo the experiment. Unfortunately, when we redid it, same result as the first. So as a scientist, we were asking ourselves, what did we do wrong? One thing could have been when we extracted the DNA the first time, we had a small population of mutants. So whenever we redid the experiment, we probably used all the mutants that were left, so we just had wild type left over. Another thing is we added, again, I'm going back to the Nuclein-3. We added Nuclein-3 to the cells in hopes that it would kill most of the wild type, which if you have questions on how that works, you can definitely ask me at the end. And so what we did is we increased, which going back to the future directions, we increased our Nuclein-3 from 10 micromolar to 15 micromolar, and hopefully it kills the majority of the wild type cells, leaving us with more of the mutants. And we're possibly looking into a nanoparticle collab with this lab that is doing more on the chemistry side. And then we have Western blot. We're going to do a Western blot because that should be more sensitive to see if we have the full protein or not. And then we're going to, whenever we do knock it out, that would be great, we're going to do some clonal selection to isolate and select single clones. We're going to grow and expand those clones. And then once we do that, we're going to, again, do the PCR to validate that we have that knockout. And then we're going to do something called cryopreservation, where we're going to put them in liquid nitrogen to store them. And then whenever we take them out of nitrogen to thaw, we're going to use them for experiments. Now, for me, when I shadowed Dr. Gupta, we talked a little bit about epigenetics, which is something that I'm really curious to see. Even if a patient has 2 functioning copies of TP53, is it being methylated? Is it being silenced when it's not supposed to? Or is it being activated when it's not supposed to? So I'm very curious about that. And I'm also curious to, like I said, isolate these models and introduce DNA damage to see which of the following would be the worst case scenario for a patient. And then we're going to do some transcriptonic validation, which is RNA sequencing, sending our cells to get sequenced, and hopefully making sure we have the right OCI-AML 2 and 3 cells that got TP53 knockout. That is the end. I just want to thank the School of Medicine for providing me this position to work as a student undergraduate research. And I want to thank the MD-PhD program for setting this up, along with the Chancellor Science Scholars program and the Cancer Lineberger Center, as I love going to Marsico every single day and seeing my wonderful labs. And again, I want to thank my lab members who came to support me, and my PI who actually gave me this opportunity even though I had no prior research experience. And so that is all. Thank you. Any questions? Yes.
Really awesome presentation.
Thank you.
So earlier you mentioned a couple of types of mutations
Yes.
that exist, like gain of function, dominant, negative. I'm curious, have there been gain of function mutations in TP53, which is known as a tumor suppressor?
Yeah, so there's actually multiple different types of gain of mutations that we're actually going to start studying whenever we get our cells knocked out. But that's a really great question. Yeah, we're going to actually use those for those as well. I think one of them is R237H is one of the mutations we're going to be looking at. That's gain of function.
So many of the point mutations are thought to have increased gain of function, but the truncating ones are loss of function.
Yes. Anyone else? Yes.
Sorry, great job.
Thank you.
So clearly the cell line that you're using has TP53 because you're trying to knock it out. Do you know what mutation they do have that has immortalized them?
That's a great question. I'm not really sure. My mentor was talking a little bit about it yesterday, but I'm going to, because I asked that question to him yesterday, which is a great one. But for the most part, I do believe that because they're leukemia cell model lines, we're using them because they do have TP53, and we want to be able to knock that out to test it. So those are like the best cells really to use in order to knock out TP53 because those are the ones that are actually like mutated, if that makes sense. And you had a question?
Oh yeah, I feel like I had to ask about the process of how you're using, I forget the name of the compound was, but to kill your wild type cells and not your mutated cells.
Oh, electroporation?
No,
Or Cas9 in the guide RNA?
No,
Oh, Noodlin 3. Oh, so actually great question. I get really excited about that. So basically there's something called MDM2, which basically, it does two things, right? So one of the things it does is it tags TP53 with something called E3 ubiquitin ligase. Well, that is what MDM2 is, but it tags it with ubiquitin. And it has a tag that allows your cells to be able to like kind of shred it and dump it out in a sense. So whenever we looked at wild type cells, they get killed because they have TP53 within them. But with our mutated cells, they don't have TP53. So whenever you're inhibiting MDM2, they have nothing to inhibit. So then that's why they survive more, if that makes sense. Yes.
And are you going to try a new guide RNA? Could it just be that?
Yes, and that's exactly. So my mentor, Dr. Martinez, I wish he was here. But yesterday, because we did a PCR again yesterday and it failed, and he was like, one thing is sometimes companies can lie to you and they send you guide RNAs, and he's like, it could not even be the right one. So yeah, that's another thing we're gonna do is we're gonna get different guides to test that out as another configuration. Yes.
Have you thought about getting it sequenced?
Yes, so we're gonna do that. The reason why we didn't do it immediately is because we realized that we didn't have much time. Sequencing takes weeks. And so Chelsea was the one who kind of told me about that, how that gets done. But yeah, so we're going to definitely do that whenever we figure out, hopefully when we knock it out. And then at that point, we'll sequence it because he doesn't want us to sequence anything that's not, we already know that's not there, if that makes sense.
Have you thought about doing, great talk by the way.
Thank you.
Have you thought about doing qPCR to see if, yes, maybe they have TP53 gene, but are they actually expressing it?
Yeah, that's a great thing. So I asked him initially if we could do a Western blot, but apparently he said PCRs is more sensitive, which is why we did that first. But I think that's another method that we can add. So yeah, I appreciate that.
All right.
Thank you.